You know, when we talk about treating a chronic disease, especially something like kidney failure, it's usually all about damage control.
Right. It's managing the symptoms.
Exactly. We're trying to pump the brakes, maybe steer the car away from the cliff edge, just manage the crash. But what if you could just reach over, turn the engine off, and just get the driver out of the car?
That's a great way to put it. That's really the shift we're seeing in precision medicine. We're going from managing the downstream chaos to actually targeting the upstream driver.
The root cause.
The root cause, exactly.
And that's exactly what we're diving into today. We're looking at IgA nephropathy, a leading cause of kidney failure, and a brand new phase 3 trial report on a drug called Sibeprenlimab.
It's a mouthful, I know, but it's a name you're going to want to remember. The report is called Sibeprenlimab, Efficacy and Safety in IgA Nephropathy Therapy. And honestly, the data in this interim, look, it's the kind of thing that really makes you sit up straight.
Sit up straight is expert speak for this is a big deal.
Yeah.
We're going to unpack why this monoclonal antibody, which targets a protein with the surprisingly friendly name APRIL, might be a total game changer. But first, let's set the stage a bit. What's the actual engine driving this disease?
Well, the engine, in this case, it's all about a very specific, flawed molecule. We know IgA nephropathy is an autoimmune disease, but it's not just some, general inflammation.
Right.
It starts with the overproduction of something called galactose-deficient IgA1.
The Gd-IgA1. That's the antibody that's missing a sugar molecule, right?
Precisely. And because it's missing that sugar, your own body sees it as foreign, almost like a virus. So your immune system makes autoantibodies to attack these misfit antibodies.
And they all clump together.
They form these large immune complexes, and those are what gets stuck in the kidney's filter.
Okay, so the complexes deposit, that triggers inflammation, scarring, and then eventually the kidneys just stop working.
Exactly. So the real driver isn't the kidney damage itself. That's the result. The driver is way upstream. It's the production of that bad IgA1.
So if Gd-IgA1 is the problem, where does this protein, APRIL, come into it? Because that's the drug's target.
Right. APRIL stands for a proliferation-inducing ligand. It's a cytokine, a signaling protein, and its job is to tell your B cells to mature and pump out IgA antibodies.
Oh.
The thinking for a while has been that patients with this disease have elevated APRIL levels, which is what's driving the overproduction of the bad IgA1.
So APRIL is basically the foot on the gas pedal.
That is a perfect way to put it. More APRIL means more Gd-IgA1, which means more kidney damage.
And that's where Sibeprenlimab comes in. A humanized IgG2 monoclonal antibody. Its only job is to hunt down APRIL and just take it out of the equation.
It binds to APRIL and neutralizes it. The logic is, it's really elegant. You block the signal, APRIL, you stop the production of the pathogen, Gd-IgA1, and you should stop the kidney damage.
In theory is always the scary part, right?
Yeah.
We've seen a lot of elegant theories fall apart in phase 2.
We have.
But this is a phase 3 trial, so let's talk about the design. Was it a robust setup?
Oh, extremely. This is the gold standard.
Really?
It's multi-center, double-blind, randomized, placebo-controlled. They enrolled 510 patients, split them 1-to-1. Half got the drug, half got a placebo.
And what was the dosing like?
400 milligrams. It was a subcutaneous injection every 4 weeks.
You know, that's interesting, the subcutaneous part. It's not a long IV infusion in a clinic. It's a shot. And once a month sounds pretty manageable.
It really lowers the patient burden. Compliance is a huge factor in the real world, and a monthly shot is, well, it's much more patient-friendly than some of the alternatives.
Okay, so the trial is set to run for about 2 years, but we're looking at an interim analysis. They peeked at the data at 9 months. This wasn't just a safety check, was it? They were looking for efficacy.
They were. This was pre-planned. They looked at the first 320 patients to hit that 9-month mark. The goal was to see if the primary endpoint proteinuria reduction was being met.
Let's define that. The 24-hour urinary protein to creatinine ratio. We know proteinuria is just the kidneys leaking protein they shouldn't be.
Correct. When those filters are inflamed and damaged, protein spills from the blood into the urine. And reducing that leakage is the primary goal because it correlates so strongly with long-term kidney survival.
So this is the big moment.
Yeah.
320 patients, 9 months in. What happened to the placebo group?
The placebo group. Well, their disease progressed, as you'd expect. Their proteinuria didn't get better. In fact, it actually increased by 2.1% from baseline.
Okay, so the car was still rolling toward the cliff. Now, what about the Sibeprenlimab group?
This is where it gets really exciting. The Sibeprenlimab group saw a reduction in their proteinuria of 50.2%.
50. 50%. They cut the leakage in half.
In just 9 months.
That is a massive difference.
Yeah.
You have one group getting slightly worse at +2% and the other getting dramatically better at -50%.
The statistical comparison is even more stark. The geometric least squares mean difference showed the drug group was 51.2% lower than the placebo group.
And I'm guessing this wasn't a fluke. The p-value.
P was less than 0.001. You just don't see that by chance. I mean, in the world of clinical trials, this is as close to a slam dunk as you can get. The drug works.
Okay, it definitely works on the symptom, the proteinuria.
Right.
But did it work the way they said it would? Did they actually turn off the engine?
And that's the beautiful part of this report. They didn't just measure the urine, they measured the biomarkers. They looked under the hood.
So they checked the APRIL levels.
They did. At week 48, free APRIL levels in the blood were down by 95.8%.
95.8%. Wow. So the drug basically wiped it out. It did exactly what it was designed to do.
It hit the target, absolutely. But the real proof is, did hitting the target stop the production of the bad antibody? Did Gd-IgA1 levels actually drop?
Yep.
They dropped by 67.1%.
That's the smoking gun, isn't it?
It really is. It validates the whole hypothesis. We lowered APRIL, the signal, that lowered Gd-IgA1, the pathogen,
Right.
which then lowered proteinuria, the damage. It is a perfect causal chain.
You almost never see it laid out that cleanly in a clinical trial. This is mechanistic validation.
It is, and they looked at other things too, like blood in the urine, and saw improvements there as well. But that drop in the pathogenic antibody is what tells us this isn't just a Band-Aid. We're truly interfering with the disease itself.
Okay, we have to talk about safety. Usually when you knock down a key immune signaling protein by 95%, you'd expect some consequences. The immune system has these things for a reason.
That's always the big worry. APRIL is involved in B-cell survival. If you suppress it, do you open the door to infections, to other problems?
So what did the safety data show?
The overall profile was reported as similar between the two groups, but when you look at the serious adverse events, the SAEs, you see something, well, something very counterintuitive.
Counterintuitive, how? Break that down.
An SAE is anything serious, life-threatening, requires hospitalization, causes disability. In the group getting Sibeprenlimab, 3.5% of patients had an SAE.
Okay, 3.5%.
Yeah.
And the placebo group?
4.4%.
Wait a second. So the people getting the dummy injection, the sugar pill, had a higher rate of serious medical problems.
That's right.
How is that possible? This seems like a paradox.
It's what we sometimes call the safety paradox. You have to remember, IgA nephropathy is not a benign disease. It causes major problems on its own. The placebo group is suffering from the natural progression of their illness.
So the adverse events in the placebo group might actually just be the disease running its course.
That is the most likely interpretation. By treating the disease effectively with the drug, you are actually preventing the serious complications of the disease itself. So the overall risk of something bad happening actually goes down.
That is a really interesting way to think about it. The baseline for these patients isn't zero risk. The baseline is the risk of the disease.
Precisely. And in this 9-month window, the risk of untreated IgAN appears higher than the risk of the drug, which is an important thing to say. It's just a 9-month window.
Right. And no deaths in either group, which is obviously great. But what about infections? Anything pop up there?
The report says infection rates were not significantly different, which is very reassuring. But you're right to ask. We always have to watch for long-term effects when we mess with the immune system.
If you stop the bad IgA, you have to make sure you're not also stopping the good IgA that, fights off the flu.
Exactly. The study continues for the full 100 weeks, and that long-term monitoring is critical. But so far, the trade-off looks exceptionally good.
So this is an interim analysis. What's the final piece of the puzzle we're waiting for?
The holy grail of any nephrology trial, the eGFR slope.
Estimated glomerular filtration rate.
Right.
Basically, the speedometer of the kidney.
Right. Proteinuria tells us damage is happening now. The eGFR tells us how much function is left. The hope is that Sibeprenlimab doesn't just lower protein, it actually flattens that downward slope of kidney function over time.
It halts the decline.
If the final 24-month data shows that the eGFR slope has stabilized, that's what proves you're preserving the organ. That is the data that changes everything.
But, given how strongly proteinuria reduction is linked to preserving eGFR, this 50% drop has to be a very, very good sign.
Historically, yes, it's a very strong predictor. A drop like this is strongly associated with a lower risk of future kidney failure. The nephrology community is already looking at this as a potential breakthrough.
It's amazing to zoom out. For so long, the strategy for autoimmune disease was just carpet bombing the immune system with steroids and harsh drugs.
Which works, sort of, but the side effects are just brutal. What Sibeprenlimab represents is precision sniping. We found the specific cytokine, APRIL, we understood its role, and we built a missile to take out just that one target.
And the result is massive efficacy with a safety profile that looks amazingly better than placebo.
It all comes back to the biology. The biology has to lead the pharmacology. When you understand the why, the how becomes so much clearer.
So to wrap it all up, we've got a robust phase 3, a 50% cut in proteinuria. The mechanism is confirmed, safety looks great, and while we're waiting on that final eGFR data, the interim look is incredibly strong.
I'd say it's more than strong. For patients who have been facing this slow, inevitable march toward dialysis, this offers a real chance at remission. Not just management. Remission.
That brings us right back to the car. We're not just tapping the brakes anymore. We might have actually found the keys to turn off the engine.
Or at the very least, a way to take the foot right off the gas.
So I'll leave you with this final thought.
Yeah.
If we can do this for IgA nephropathy by targeting APRIL, what other drivers are out there hiding in plain sight for other so-called incurable diseases? How many other conditions are just one APRIL discovery away from a real solution?
That is the billion-dollar question. And the answer is probably more than we think.
Thanks for diving deep with us today. Keep asking questions.
And we'll keep analyzing the answers. Until next time.